about one more topic. It is gram-negative pathogenic cocci, and namely Neisseria genus. Neisseria genus is the pretty big genus, but there are just two medical important specimens. Neisseria meningitidis, or meningococci, and Neisseria gonorrhoeae, or gonococcus. Well, let's talk about both bacteria. Here you can see pictures of both of them. Here is Neisseria meningitidis, and here is Neisseria gonorrhoeae. They are pretty similar, but there are a few differences. For example, structures. Structural difference is the only one. Well, both of them are gram-negative, both of them are diplococcus. The shape is kind of coffee beans, arranged in pairs. Both of them are encapsulated, but the only difference is in Neisseria gonorrhoeae, as well as you can see in this picture, Neisseria gonorrhoeae can have pili. Well, it is a structure which helps in adhesion, in anchoring of the bacteria on surfaces. Even if Neisseria meningitidis doesn't have pili, both of them have capsule, and in some cases, capsule is making the same function as pili. It helps to adhesion. But anyways, the main function of the capsule is protection, and this protection is against phagocytosis. So please don't misuse the function of capsule. Sometimes somebody is thinking that the function of capsule is protection against factors of the environment. So it is not. It is not right. Namely, Neisseria genus is pretty sensible to the environmental factors. These bacteria are not really resistant in the environment, so the main function of the capsule is protection against phagocytosis. This is one of factors of pathogenicity of this bacteria, because encapsulated bacteria are resistant for phagocytosis. That means that when the phagocyte will swallow the bacteria will be able to survive inside. Moreover, it will be able to multiply inside the phagocyte. So this very bacteria will be not just safe, but also it will be invisible for a short period of time for immune system, this very bacteria. If there are any other bacteria which didn't succeed to get inside the phagocyte, of course, the immune system will catch this bacteria. Well, about Neisseria meningitidis, what do you need to know? That it is fastidious microorganism, and whenever you can see that the mostly comfortable temperature for a pathogen is 36, 37, 35 degrees. What do you need to know? That it is the best temperature for every pathogen which can produce a disease in human. Well, just keep in mind that every pathogen which can produce a disease in human has optimal temperature between 35 37 degrees. So also it can require some CO2 or candle jar. You can remember we was talking about such kind of creation of anaerobiosis. So if you want to remind this topic, please go to a video on the channel, which is called anaerobiosis, and you can find more information about it. So it can grow in blood agar and in chocolate agar. Well, Neisseria genus prefers this kind of culture media. blood and chocolate agar. And here you can see an example of colonies of Neisseria meningitidis on the chocolate agar. You can see that they are small with a smooth surface, gray colonies. Transmission of Neisseria meningitidis is usually by droplets, which are flying through the air because caused by sneezing, coughing, and so on. Well, they can get from the respiratory system in the bloodstream, and from the bloodstream they can get in the brain, and they will be able to produce meningitis. Moreover, they can produce septicaemia. So in some cases, it gets faster in bloodstream and can produce septicaemia. In some cases, it gets in brain and produces meningitis. Moreover, since it is an infection which produces meningitis and which is spread through the airways, it can get in the nose and through the olfactory nerves. It also could pass to the brain and produce meningitis. About clinics, its incubation is between 2 and 10 days. Symptoms could be stiff neck. It is one of the first and important symptoms of meningitis, since it produces inflammation of the brain, of the meningeal covers, and it multiplies in the cerebrospinal fluid. It produces tension, inflammation, and it will increase tension of cerebrospinal fluid. So it will press more centers and it will produce stiff neck. Of course, it will produce extremely high fever and sensitivity to light, confusion, headaches, vomiting. All of them are associated to pressure on more centers in the central nervous system. So all of them are signs of infection within central nervous system. Less common, but even more severe, it can produce even death, is meningococcal septicaemia. So it can produce haemorrhagic rash. It will spread all around the body. It can produce circulatory collapse, and in very short period of time, it can lead to death. So even if the disease is diagnosed early, even if the treatment is started on time, a lot of patients, usually around 10%, between 8 and 15% of patients, are dying within 24, 48 hours after the onset. About the lab test, the gold standard is isolation of bacteria. So usually we are taking either blood or cerebrospinal fluid from the patient. We are taking it from culture. Sometimes we can take other fluids in dependence on symptoms, but usually it will be blood or cerebrospinal fluid. And we'll make gram staining of cerebrospinal fluid. Why namely cerebrospinal fluid? Because normally it isn't supposed to have any other cells. So if in blood you will find a lot of different cells, and it will be complicated to stain blood by gram. Cerebrospinal fluid is not pretending to have any cells, so you will find there just bacteria. The first sign that you have, namely bacterial meningitis, but not viral, for example, the cerebrospinal fluid will not be transparent. So if we have viral meningitis and you will make cerebrospinal puncture, the cerebrospinal fluid is supposed to be transparent. If there is bacteria, it is bacterial inflammation. The cerebrospinal fluid will not be transparent. It will have certain turbidity. So this is how you can make a difference between them. If the cerebrospinal fluid is transparent, highly likely that you will not find there any bacteria. And other tests are serological tests, ELISA, for example, and PCR. The treatment is to be started as soon as possible, of course with antibiotics, but only after the spinal punction. Well, uh, uh, even if you are giving treatment, you should understand that meningococci will stay alive a little bit longer in cerebrospinal fluid than in blood, because you are introducing the antibiotic intravenously, so it starts working firstly on bloodstream, and moreover, as well as you can know, the central nervous system has a specific hematoencephalic barrier, which filters a lot of substances, including drugs. So the antibiotic will get harder in the central nervous system and in the cerebrospinal fluid. And the treatment is to be applied with penicillin, with ampicillin, and so on. Regarding Neisseria gonorrhoeae, morphology is similar to Neisseria meningitidis, but the only difference in structure is that gonococcus has pili. Well, it is able to make adhesion. And transmission, if in meningitis, transmission is through the air. In gonorrhea, the transmission is sexual. There are a lot of discussions about transmission via using of towels, sponges, and so on. I don't know how relevant this method of transmission. Of course, technically it's possible, but the question is how you're supposed to use that towel, or how you're supposed to use that sponge, or whatever it is, those objects, in order to get this infection. So it can be supposed that usually people are just using such explications of how did they get this infection in order to avoid some uncomfortable situations, or in order to avoid saying the truth, as well as you could know the expression that everybody lies. So it could be applied for this case. However, nobody will deny that main way of transmission is sexual transmitted disease. Well, this disease is transmitted sexually. Regarding clinics, there are several nosological forms. On the first place, of course, we have genital infections, and as a result, there will be secretion of pus from genitals. Namely, usually it is morning droplets of pus. By the way, the name of gonorrhea appeared from gono, which means semen, and rhea, which means flowing. Well, in the beginning, before this disease were studied good enough, people were thinking that gonorrhea is flowing, is running of semen, since it is mostly spreaded. Symptomatic form is mostly spreaded in men than in women. So Usually men has such symptoms that in the morning they have running of pus, which was people were pretending it is running morning running of semen. So actually it is a pus. This is why this name appeared, and it remained actually till nowadays. Well, on the first place we have genital infections, genital inflammation. On this next we have proctitis and throat inflammation. In case of practice of different sexual relations, it will affect different organs, either even rectum, either throat. Also, it can happen conjunctivitis, but neonatorum. That means it can affect newborns. And namely, since genital infection usually passes in women without any symptoms, the infected woman can be even pregnant and she couldn't know about that. But during giving birth to a child, the mother can infect the infant, can infect the newborn with gonorrhea, because the newborn is passing through the birth way and has direct contact of the face with mother's vagina. So in this way, the child can get infected with with this pathogen. And in chronic gonorrhea, if it wasn't treated, or if the disease passed without any symptoms and some bacteria remained in the body, it can lead to inflammation of big joints. So how you can find that this arthritis is related to gonorrhea or not? Just by precedent, by previous symptoms, or by knowing of relations the person had. So, are women more dangerous or not in this infection? Well, what do you need to know about epidemiology of this infection? If the person doesn't have any symptoms, the person doesn't know that he or she has the disease. And as well as sexually transmitted infections are transmitted usually among people who are having multiple partners. If the man which has multiple partners will get this infection and will have some symptoms, of course, usually he will take the treatment. But as well as we know that women very often have asymptomatic form of this disease. She doesn't have any symptoms and still she has multiple partners. Of course, she will not know about she is infected, and she will continue spreading this infection. So from this point of view, from this in this very case, women usually are more dangerous epidemiologically than men in transmitting of gonorrhea. Well, as a lab test, we will apply the same types of investigations which we are using in Neisseria gonorrhoeae, in Neisseria meningitidis, but the only difference, we will take specimens from another place, independent of which area is affected, which organ or system is affected. More complicated in chronic gonorrhea, because in chronic gonorrhea the only symptom is inflammation of big joints. So we don't have any production of pus. If we don't have production of pus, we don't have access to bacteria. So how can we make investigation in chronic gonorrhea? Well, first of all, we can make serological test. We can take the blood for finding of antibodies, or we could use the fact we know that Neisseria gonorrhoeae is pretty sensible to spicy food. So if person will take some spicy food or will take some alcohol, it will be kind of trigger for Neisseria gonorrhoeae, and it will produce acutization of the infection. And in this way, you will be able to produce some pus, and some bacteria will be found. And of course, if first two ways we will not apply a medicine, we can apply hot water bottle. It will produce the same effect as first two ways. Regarding treatment, we have antibiotics. We will apply ceftriaxone intramuscular, and we will apply either azithromycin, either doxycycline, per oris, so intraoral. And as well as we can know that the majority of people are who are getting sexual transmitted infections because of practicing of sexual relations to multiple partners, highly likely that the majority of them will not have just gonorrhea, but they could have other sexually transmitted infections. So in this case, of course, we are supposed to apply the treatment for different infections. For example, in some cases, there are fungal infections, and some of Neisseria could hide inside of such pathogens. So in this case, we will need, of course, to apply complex treatment for this infection. Well, that's all for this topic. Thank you for your attention, and please feel free to give me your questions.